Hello, this is Mrs. Murdock, and I'm going to be talking about different types of ways to make enzyme graphs and to interpret them. Specifically, this is a help video for you if you are working on the enzyme lab formative activity in AP Biology. So we're talking about enzyme graphs and what they mean, really. That's and their analysis. Okay. So you guys did two experiments. You did an experiment, we'll just kind of focus on A here. You did an experiment where you varied the substrate and kept the enzyme the same, fixed, or finite. There was a finite amount of enzyme. Finite. That's a good word to use, okay? And when you actually did your graph, you graphed the substrate concentration down here. Your substrate concentration, that was hydrogen peroxide, right? And it only went up to 3% because you can't buy stronger than 3% in the store. That's the only reason why it didn't go higher than that. But it gave us the data that we needed. And then you actually calculated an actual rate of reaction, right? Which was how fast those disks went up and down in your test tubes, right? And your line, your line kind of looked like this, right? I mean, everybody's lines look like that. So interpreting this line, pretty simple. I didn't ask for error bars here. I wasn't asking for any kind of thing like that. It's a pretty obvious claim. I'm not going to help you with that. I think you can figure that out. But the reasoning part of this has to really show some detailed, in-depth understanding. And that's what I'm going to try to guide you with here. So if I go over here and I look at this point on the graph versus like this point on the graph, right, where the substrate concentration up here is like 3% and down here it was, I don't know, maybe 0.5%, I don't know what it was. So which part, at which point is the reaction going faster? I know you don't want to be on film, so I'll just say it. So yes, here the reaction is happening faster. Literally that point means the reaction's happening faster than it is down here. It's going more slower at this point. At this concentration of substrate, it happens more slowly than when you add more and more substrate and it goes faster. That might seem like a simple thing, but now you need to explain that, right? So claim, I have no doubt you have that part. Evidence, you can do that, I'm sure. You can make it quantitative even. I think you can do that just fine. It's the reasoning that I'm really going to be a stickler about and looking for some detail and some in-depth understanding. So you need to be able to use that list of enzyme terms that you wrote down from that pair deck the other day. All of those terms. And you need to also think about that video that I showed you that literally showed you the inside of an active site and what was going on in there. And what an enzyme really does to a chemical reaction. And there's also one other thing that's in there that, that you may be wondering about. It's this thing called the kinetic molecular theory. So this is a pretty good definition here. If I read this to you, it explains the states of matter and is based on the idea that matter is composed of tiny particles that are always in motion. This theory helps explain observable properties of solids, liquids, and gases. And almost always when you look up kinetic molecular theory, they talk about gases a lot. But this also applies to liquids. Molecules are moving in liquids. They're always colliding against each other. Not quite as dramatically as in a gas, but there are molecular collisions to consider. So you're talking about, pull this down here. the term I really want you to think about is that we have molecular collisions going on of enzyme molecules, that's one type of enzymes, and substrates. Okay? So think about that, the fact that the more particles you add, right? The more molecules you add in there, the greater the amount of collisions there are going to be between them. That just makes logical sense, doesn't it? If you add more molecules, there'll be more collisions. There'll be more of a chance of substrates bouncing into active sites and reacting, literally, right? So, the more molecules you add, you increase collisions and increase chance of reaction. So that's sort of what we're talking about here. And when we're talking about enzymes, that's a good thing, because the other way that you can increase collisions is to heat it up. And we can't put a living cell in boiling water, right? You can't heat up living cells. It's not good for them. But you can add more enzyme particles or more substrate particles, and that increases rate of reaction. So you have enzymes. I like to make it a little Pac-Man for enzymes. Let's say you have enzyme particles, and let's say you have a finite amount, right? The yeast that you put on the surface of your filter paper was providing a certain amount of active sites. A finite amount of active sites. You didn't vary that in A. And then you're adding, maybe you only had a little bit of substrate at first. Maybe you just had that. Right? There's your substrate. And the substrate fits into those active sites, and if there's only a little bit of substrate, it's all going to go in there into the finite active sites, and it's not going to go that fast. Right? But the more substrate you add, the faster and faster the reaction is going to go. That should make sense to you. That's what you need to explain. Okay? And here's another thing, because there are some questions down at the bottom of the formative that I don't want you to get confused about. Our lab was a straight line, wasn't it? Looks like it's just going to go on forever. But think of it this way. What if, same thing, you have rate of reaction here, and you have substrate concentration here. Concentration. And what if here's 3%, which is as high as we were able to go, right? All right. What if we could get up to, I don't know, 50% hydrogen peroxide? That'd be some pretty intense hydrogen peroxide. You wouldn't find that in a store. Okay? What do you think that curve would look like? If you have so much substrate over here, and still only a finite number of active sites to receive it, what's going to happen to this curve? Is it going to keep going up and up and up? It's going to plateau at some point. There's going to be a point where the active sites get saturated. Okay? So active sites can get saturated, and that doesn't mean the reaction stops. It just means it can't go any faster than that. It's still going that fast, whatever that rate is, but it can't go any faster because that's all those active sites can handle, that finite amount of open mouths waiting for the hydrogen peroxide, right? Okay. All right, so hopefully that helps you think about that. The last thing I want to talk about is that there is in fact another way to graph enzyme data that we didn't do, and I just want to make you aware of it. So in some labs that we've done in the past, another way to graph enzyme catalyzed reaction data. Is, let's say that you don't measure the rate at all. You just measure the disappearance of the substrate and appearance of product. A lot of labs actually do that. Some of the high school labs we used to do, we didn't measure rate. We just measured how much product was produced per minute. So you had time down here, the time of the reaction. And then here you had appearance of product molecules, concentration of whatever the products were. Maybe in this case it would be oxygen bubbles. And that kind of graph, if it's happening, often it was happening in some kind of a beaker, right? So a finite space, you put some substrate in, whatever it is, and you already have enzyme in here that's finite. It's there. And the reaction fizzes up and then goes to its endpoint, right? And you measure the amount of product as it appears. And it winds up looking like this. Which looks exactly like the curve on the other graph, doesn't it? Yes. Except, look at the x and y axes here. Appearance of product is not rate. Time is not substrate concentration. So you have to realize what's actually being measured here. So in this case, down here and up here, let's say those 2 points, where is the reaction happening more quickly? Is the rate faster here, or is the rate faster here? Yeah, so here it's faster. Yes. It's faster here because amount of product, let's say, I don't know, bubbles. Amount of bubbles per minute, right, whatever that is, that's slope. The slope of the line equals the rate of the reaction. And we'll actually talk about that for a lot of experiments, is when you have anything happening over time. Anything that happens over time, if you take the slope of the line, that gives you the speed of that thing happening. So in this case, it's happening faster here. There's more bubbles here per unit time. And then up here, no more bubbles. Reaction has stopped. So watch your X and Y axes and just be careful of that. Okay? All right, that is all I wanted to say about enzymes and ways to graph enzyme labs and how to explain and think about that kinetic molecular theory, molecular collisions. It's now up to you to finish your formative labs together. And I hope that was helpful.